Hello,
Hello,
how are you?
I'm here.
Where are you located?
My son's house,
Okay, where is that?
near Winnipeg.
Okay, yeah.
So I'm near Winnipeg now instead of off in the boonies.
Oh, so welcome to the meeting. Again, we're working on our GSoC proposals, working on different things, and everything's coming along. I'm actually working on a couple papers, getting those into a volume with Dick and with Leywith, so that's good. So if you're not familiar with the GSoC schedule, if we look at the schedule, the deadline for proposals is March 31st. So that's coming up in less than 2 weeks. So I guess, if you're applying for GSoC, you should start to get your proposal written up. Now, if you want to know how to write a good proposal, my suggestion is, of course, we've had people presenting in the meetings last several weeks. So if you have presented, you have, I think, a good focus on what your proposal will look like. You know what the question is that you're answering that we have in the in the in the call for proposals. Then you have to write your kind of answer to that and present what you've basically put into your PowerPoint notes. And then you need to have a schedule. And so the schedule is a list week by week of what you plan to do. And so you go to the schedule for, they have it on the GSoC website. Of course, you have the 2 weeks of community time, community period, which we take fairly seriously in this org. And then you go week by week from the end of the community period to the end of the coding period and say what you're going to do. Now, you have to be careful because sometimes you can propose too much, sometimes you can propose too little, and sometimes you might propose things for which you can't actually complete those things. Like you might run into problems, you might not be able to implement something. So, what is your plan for getting around that? What is your plan for not losing time because you are trying to implement something? And so that is what I'm looking for. And it's a good, I think, testament to thinking about what you're going to do and having a good command of scheduling and of timing. It's an important part of project management is figuring out how long things will take. Now, that being said, do you have to get the schedule right? Not at all. You can get the schedule, you're not going to get it right the first time. You'll have to sometimes rescope your project, and that's okay because this program is designed to help people work out the scope of their project as they go. It's just that you don't want to get in the situation where you kind of rescope it on purpose. You kind of don't make very good estimates, and then you're doing all sorts of things that aren't in the schedule, and then sooner or later, or I guess sooner, you end up sort of with scope creep, and you're trying to do all these different things, and none of them really lead to your success. So that's what my suggestion is for proposals. Just be very clear, very focused on your, what you're going to do week by week for implementing the thing that you're kind of worked out already. You worked out the big picture. Now you can focus in on the details. And that's what the schedule is for. Okay, so that's a few words about that. So those are due on March 31st. Okay, so it looks like we have one instance of Susan, one instance of Morgan, and 3 instances of Shreyas. And
yeah, I don't know actually why my connection was going. So
yeah,
I went and came. So like the other instances didn't go off. I don't know why that's happening. But yeah, now my connection is stabilized.
okay. Well, yeah, that's good. So I guess Susan. So last week I wasn't here, and Susan was here. and she shared with people her presentation. And I found that interesting. She sent me the presentation. I did find one graph interesting, or well, I found a lot of graphs interesting, but one graph interesting. So is this graph here, modeling of tissues. So this is I guess from Kim et al. 2020, engineering and modeling of multicellular morphology and patterns. It's in current opinion in genetics and development. So this is like all the different ways that people model tissues. You have Monte Carlo methods. So you have discrete models and you have continuous models. And then you have design aids and heuristic models, and then physical models. So I'm not. No, I mean this looks good, but I'm not sure if this is exactly the way I would do the typology. Anyways, the discrete models are Monte Carlo, particle based, and lattice. Okay, so the discrete models are things like CompuCell 3D or like rulology in cellular automata, those sorts of things. You can model emergence, you can model stochastic processes, you can model neighbor interactions. Then you have the actually you have two here, continuous models, stochastic reaction diffusion. So it's like a reaction diffusion model where you have particles. It's like diffusion limited aggregation, or sometimes just like a chemical morphogenesis process where you have this stochastic process and you have diffusion across the space, and you have reactions where they collide or where they meet in gradients. That's one approach. And then there's the discrete plus continuous, which is vertex and reaction diffusion. So taking reaction diffusion and turning it into kind of like a quasi network. So you have this little star here. It's like a starfish maybe, where they're modeling like form instead of having a big blob. They're modeling like specific form, directional changes in form. And then agent-based and diffusion, which is where you have these agent-based models. And this is more your classic diffusion limited aggregation. So diffusion limited aggregation is where, like, if you want to grow a snowflake from a bunch of particles, you can use the diffusion limited aggregation approach, which is basically if I take a bunch of particles and I sort of throw them randomly across the field, and then I do this enough at a high enough rate so that particles will hit each other. And when they collide, they kind of become stationary in that place. And then eventually you start to get clusters form, and then clusters of shapes. And people have done this to generate snowflake patterns. And it's really interesting how kind of what kinds of pattern formation you can get from that. Then that's discrete plus continuous. Then the design aids. This is evolutionary algorithms where they're modeling a tissue using. This is I think Kriegman 2020 where they're modeling in a cube format. They're evolving shapes from cubes that are stuck together. And then they have finite element. This is where you have these shapes that are built from finite element analysis. And then there's heuristic models, probabilistic models, where you have your cells and your neighbors. So this is a cellular automata neighborhood where you just, each cell takes the state of its neighbors and makes a decision based on what the state of its neighbors are. And then you have physical models where you have swarm robotics. And this is where you have these robots where they, the pieces come together and they follow the rules of sort of swarm intelligence, which is this collective emergent behavior. So I like that figure because it kind of goes over a lot of different methods and how they kind of fit together and how you might use them under different conditions. So I don't know, Susan had anything to say about that figure. I didn't see the talk firsthand. I did see the slides.
I think I just went over it just briefly.
Okay.
than you did. So that's fine. That's just, it kind of gives you an overview of what's going on.
Yeah.
Like so many models out there.
Oh yeah, yeah. The model zoo, as the physicists call it.
Yeah. Anyway, I used finite element analysis on structure. So
Okay. Yeah.
pure engineering on it. So
Thank you. Shreyas, are you there?
Yeah, I'm here.
Hi. Did you have anything to present this week or not?
Yeah, I do have something to present.
Okay.
I'll start the presentation.
Yeah, yeah. Go ahead and share your screen.
Yeah, just midway making a presentation. Okay, so this week I did talk to Mehul regarding as to what my progress is and how can I go ahead. So he also stated that you can look into the OD part as we talked regarding the OD part can be interesting, and he also told me that he can talk on lineage as well and try to model using hypergraphs as to how lineages can be used and how clusters of neurons would move using a hypergraph. Right. So he asked me to think in that direction as well. So the preceding 2 weeks I did go into that direction and tried to validate if it's actually useful and how can things be done. Okay, so I'll just go over it. Yeah, so this is interpolation of connectome, taking into account the lineage. So up until now, lineage was not explicitly taken into account. The methods which I was implementing did implicitly or like indirectly take into account the lineage, or we found like the lineage matters, but explicitly lineage data was not taken into account. So yeah, why lineage matters? So lineage matters for neural development modeling because so the each neuron I is embedded at position Zi such that in a previous approach is what I'm telling. So it was such that the Fermi direct probability. So this is where each of the neuron was embedded in my hyperbolic space, right? Hyperbolic space, and they were just embedded individually. The lineages were not taken into account. So they were embedded such that the Fermi direct probability best matches the true adjacency matrix. Now, this approach was powerful, but as it recovers the strong anti-correlation between hyperbolic radius and degree. So what this means is that how if a neuron has a high degree, so it has small radius that is at center around the center of my Poincare ball or Poincare disc, whatever we take. Because its Pearson radius R was approximately minus 0.75, which is a strong anti-correlation. But it treats all neurons as exchangeable priors to observing the connectivity. So basically, two neurons with identical degree distribution but receive identical priors. But what are we ignoring here? The blind spot is developmental history in C. elegans. So two neurons can have a near identical adult connectivity yet arise from completely different progenitor cells. Meaning, like conversely, neurons from same progenitor often share connectivity signatures. So what does this mean? Is like if a neuron arise from some lineage and so a neuron B from another lineage, they may have similar degrees, but they can have completely different characteristics as their progenitor is different. But if some neurons have same progenitor, they share connectivity signatures. How do they grow and how connections happen that are not predictable from degree alone. So degree is so we are having some basically we are ignoring the lineage as of now. So what my hypothesis is, the lineage hypothesis is that neurons that share a common ancestor cell in the embryonic division trees are not independently distributed in hyperbolic spaces. Instead, they form clusters. So like each neurons arising from particular cells would form a cluster, which is called hyperedge. I'll go into what hyperedge is. So that moves. So that will move collectively through the Poincare ball during development. So the spread of the lineage cluster is a geometric proxy for degree of fate diversification within that lineage. So basically, what this hypothesis means, I'll just summarize. If I had to put a one-liner, is that neurons come from any single lineage would tend to cluster and move together in the hyperbolic space. Right. So now, what is my data? So data is my lineage tree and connectome. So connectome data is something which I have been using for a while now. The Whitwittel paper. This data set is a lineage CSV which provides 642 rows. So each encodes one cell division. So there's a parent cell, like one column is for parent cell, and for daughter 1, daughter 2, time of birth and position. This is what my data is. So that tree has 12 depth 12 from T0 zygote within 741 terminal leaves. So this is how. So I'm just covering the nomenclature here. So here A, I guess we are familiar with this. Like A is anterior, P is posterior, A is left, right. So how the division occurs. What is dorsal and what is ventral? So therefore the name literally, so it decodes, it corresponds to the name encodes how the division has occurred. And it's all history, the whole history of the cell, how the division occurs. Right. So I have done. So now what task is we need to do the mapping from lineage tree to connectome. So connectome naming is different, lineage tree naming is different. So we need to do mapping. So as to how they relate. So I used a data set here called Bombay's data set, which is the mapping. So this data is from Sulzon's paper. So we explored the Sulzon naming convention to construct a robust 3-tier assignment using Bombay's. So this is the assignment which happened. So 40 were unknown. So this is the lineage so this is the tier 2 lineage. It has a depth is 12. So I used like depth 2 as my lineage indicators. And then I went ahead. So this is my depth 2 lineage. And I'm mapping each of my neurons. So this is the naming in the Wittfiliate paper, like ABA, RMD, RI, and so on. So this is the mapping which has been done. Now, like we changed from graphs to hypergraphs. So as of now, I was using graphs. So what is the standard graph edge? It's like it encodes a pairwise relation between two neurons. But the cell lineage relationship is not pairwise. It's a many body constraint. So all neurons in a lineage group simultaneously share a common ancestor, and they collectively respond to the same progenitor level determinant. So like all of the lineage for the whole tree would have a similar characteristics. So the hyperedge generalizes a graph by allowing edge to connect to like it connects to many number of nodes. Basically, it allows many to many connections. So each lineage group is a hyperedge. So all the lineages form hyperedge for each lineage. So since this is a binary tree, so as you know, like this, of course, the division is a binary tree, like 1 to 2, 2 to 2, 2. So we use a concave as a perfect model to use this. So like this is mathematically established that for binary trees, concave ball is a perfect model, and I proved in previous presentation as well that how concave ball is valid here. So to compute velocities, now why do I need velocities? I'll just come to this in a while. To compute velocities, we perform linear operations on manifold. We use the exponential and logarithmic map at the origin. So what is basically exponential logarithmic map? Basically means the tangent space at origin. So I have a pointer ball. So if I have to take velocities at any point, again, I found what is velocity here, but if I have to take velocity, I'll just take the tangent space at the ball. So say if I have a ball, I'll just take tangent space here to see velocity here at this point. Right. So this is what, so we shifted to hypergraphs now. So now collective motion. So as I said previously that the neurons arising from same lineage collect to move together. So what does this mean? Is that we say the lineage group E exhibits a collective motion in the hyperbolic space between stage T and T1. When do we say is when the centroid ZD follows a smooth geodesic rather than a random walk. So it follows a smooth geodesic on the concave disk. The spread. Now what is spread? I'll just come to this. I've written here what is spread. The spread 6 sigma 2e changes slowly and smoothly rather than fluctuating randomly. That means that okay, it's following some trajectory. And the direction of individual neuron drift correlates with the direction towards a lineage centroid. So if all three hold, the lineage group is behaving as a geometric unit. The neurons are aware of the family memberships in a sense that the trajectories are constrained by the collective position of the group. So this means again, what is geodesic spread? So just not to go into the exact mathematics of it, like I have gone and coded it up, but not to go up right now. Geodesic spread, we can assume like it's a variance on the manifold. So we know what the variance is in our Euclidean space or general probability terms, statistical term, what the variance is. So that is just the variance on any manifold is called on manifold. So here it is the hyperbolic space. It's called a geodesic spread. So now come to this graph. Here I have just modeled how my spread happens over various lineages. So as you can see, I have taken 3 lineages here. The first one was 0, so we didn't map any neuron to that lineage. So we can see how the geodesic spread varies. Other than the seventh developmental stage, the developmental stage corresponds to Wittfiliate paper, where it was like 1, 2, 3, and we need to interpolate across 1 to 8. So we are corresponding that, getting that data and see how the geodesic spread is going for various lineages, right? So yeah, I'll just interpret this data in a while. Yeah, the lineage direction as a collective force. Now this is where the velocity term comes in. So the key quantity linking the hyper structure to OD interpolator. Now just to give a larger sense as to what we're doing, everything which we are doing is to take um, uh, parameters for my OD. So these all will build up my parameters for the OD. So OD is the central unit of my model, the neural OD, and we are building parameters for it. So lineage is also a parameter. So the key quantity linking hyperstructure to the OD interpolator is the lineage direction vector. Right. So this is my direction vector. Again, the mathematics of it aside, but so I'll just tell the intuition and interpretation. So this is the unit vector pointing from neuron B's current position towards its lineage centroid, computed in the tangent space at origin. So geometrically, it basically represents the direction the neuron should move if it were to converge towards its group. So if we assume that the neuron is converging towards its lineage's group, then this is the direction which it should go. So lineage distance represents how far. So now there's another term called lineage distance. So this was lineage direction vector. This is now lineage distance. So lineage distance represents how far the neuron currently is from its group centroid. So together, these two terms encode a three-dimensional vector that the OD field uses as its input. Right? So OD can learn using this information. So we are giving the OD this information, and then we assume that it should learn. Like, let's see. Now this is the result one. So now this I have set the premise of whatever I'm doing as to why I'm doing and how it can be useful. Now we have to validate that if this actually makes sense. Right? So result one. This is statistically the distinct hyperbolic region. Yeah. So this is the Kurzweil-Wallis test on spread distributions. So what is basically does? So this basically tests whether the samples CT are drawn from the same distributions, right? So I'll just just move to this PDF here. So this was something which I just built. So I started writing the report actually for the for GSoC. So I'll just go to that section where the results are there. So again, here mathematics are stated quite deeply. So if you can see, I've stated here mathematics of everything and the proofs and all. But in the PPTs, I'm just pointing out the results. But in the report, I include mathematics as well. Yeah. So just to go to the quantitative results and validation. So my result one states statistically distinct hyperbolic regions. Right. This is what we're testing. So yeah, this I showed. I interpret these values. I'll just first tell what the test is. So the Kurzweil-Wallis test is a non-parametric analog of ANOVA. So what it means, it tests whether the samples. So these are my spreads for each of the lineage, right? With time. So are they drawn from the same distribution? So rejection at p-value. So I'm assuming like there's a statistical basis as to p-value. I'll just give out what a p-value is. So the p-value means the spread distributions are significantly different. So this means that okay, they are not overlapping in their trajectories, and they are significantly different distributions. So these are drawn from a different distributions since the p-value is very low. So lineage. So what the result is? Result is a lineage group occupies statistically different regions of hyperbolic space, and lineage identity is a significant predictor of hyperbolic position. So now we know that okay, in the hyperbolic space, the lineage actually differentiates its position. So this is what this number tells. So this is test one. Now result two is the test. It shows the fate diversity predicts spread magnitude. Okay, so at adult stage, my spread are spread across the what is it, lineages. So these are I would say these are the founders. Like I quote, these are the founders like ABP, ABA, C, and so on. So this ordering exactly matches the known fate diversity ranking. So I just looked up through the biological papers, like the one from which I obtained this data. So this diversity is matching with the theoretical one. So ABB produces neurons of all functional classes. ABA produces a narrower range of primary anterior neurons, and C produces exactly two neurons with defined non-overlapping roles. So the quantitative ratios reflects approximately 40-fold difference in neuron diversity. So this tells the fate diversity which is projected through the magnitude of my spread. Right. Spread again, just to recap this graph. Right. So now result 3, spread decreases post synaptogenesis. So yeah, so basic linear regression on this time spot gives So table 4, yeah. So this is my slope and p-value. So this basically tells, so the negative slope confirms the contradiction towards linear centroid after peak synaptogenesis. So both ABA and ABP show significant post-peak decrease consistent with circuit stabilization after peak synaptogenesis. So again, I'll just tell what does this mean as to circuit stabilization post-peak synaptogenesis of stage 3. Let me just see if I have that somewhere. Just a second. Let me just see if I have that graph somewhere where I show. Okay, never mind. I'll just tell. I don't know exactly where that graph is. I'll just tell what I mean. So this means that so my as I stated in my previous presentations, so my radius which I'm quoting here, the hyperbolic radius, it is small initially. It maxes out around stage 3 and 4, and then it decreases a bit. So as it maxes out around stage 3 and 4, the quantity in which it decreases is of course less than 1 to 3. So if I take the difference in radius of stage 3 and 1 and 3 and 8, of course the difference between 3 and 8 will be lesser than 3 and 1. But essentially my point is that it maxes out around stage 3 to 4. So this is what basically it means, that so after stage 3, circuit stabilizes till stage 8, and this is followed by my the spread the spread decreases post synaptogenesis, and the result 4 is that hyper-edge smoothness decreases monotonically. So basically these are my values for smoothness. So basically how much does the embedding respect linear structure? So smoothness means geodesic distance of each neuron from its linear centroid. So I'll come to the conclusions for this. So this table 5, the hyper-edge smoothness across developmental stages, there is a monotonic decrease from stage 3 to stage 8, confirming progressive lineage geometric coherence. So ST peaks at stage 3 and then monotonically decreases to stage 8. So this is the cleanest signal in the entire analysis. So with a negligible uptick at stage 6, but that is like fine because a new motor neuron addition happened at stage 6. So yeah, this basically the interpretation is that from stage 3 onwards, the neurons progressively cluster closer to their lineage centered in hyperbolic space. So the embedding becomes more lineage coherent as development proceeds, and this is a continuous measurable geometric signature of neural maturation. So this again it coincides with my radius analogy which I told. I'll just see if I can pull out the graph. I'll see that later. I'll first present my analysis. So result 5 is cross-scale radius degree law. So as I specified, that degree is anti-correlation is there, specified in the start. So also holds at, so this individual this was as a whole of the neuron. Like the degree, the radius degree anti-correlation, but this same relation also holds at lineage group level. So for each lineage group, same result holds. So the mean radius and the mean degree per lineage group at stage 8 has been there in this. So the anti-correlation holds at each group level. So so the interpretation of what I meant to say is for ABA, there is a high connectivity near the center. For ABP, is a high connectivity near the center. So MS has like near zero connectivity. MS is at boundaries, and the C lineage is also near the center. Right. So this cross-scale consistency confirms that the hyperbolic geometry is capturing a genuine structural principle of connectome operating at both individual neuron level and lineage family level. So what this test basically validates is that me using hypergraphs for lineage distribution and modeling everything in hyperbolic space actually makes sense, and I can extract signals from this for my ODE. So again the center thing is ODE, and there are supplementary things which will be going input to the ODE. I'll just skip over to the alignment with neural developmental modeling. So my goal, as the original goal stated, is overarching scientific goal is to learn a continuous model for a neural circuit formation. This is dz by dt equal to this is a very simple model. So what I want to do is I want to do dz by dt equal to some function of my z, biology, and theta. Right. So such integration from any observed stage to any other accurately predicts the intervening connectivity. So biology term is crucial differentiator. As like biology term is these are the facts which I stated out a while back. as to how I'll ground my model using my biology and various papers which are stated. So, yeah, my lineage direction will be a structural force. So it will be, as I said, like this D linear equation, right? So it provides OD with information about collective position of each neuron's lineage family. So how what will the correct? So my spread as a developmental clock as to how my spread should proceed, and lineage priors. But yeah, so like just the overall thing is that uh, uh, my hyperbolic space and its the hypergraphs would provide the direction and velocity for each of the neurons in the context of how its lineage group is moving across the hyperbolic space. Right. So yeah. So any questions?
Yeah, I had a few. Let's see. Okay, well, thank you. Thank
Yeah,
you.
sure. Please tell me what
Yeah,
point
yeah.
the
Um,
question are.
so it was really good. Yeah, so one of the things I noticed was that you kind of had something like the velocity field that you kind of came up with. So that reminds me of the
Right, right, right.
yeah, the it reminds me of a Lagrangian coherent structure almost. If you think about like
Uh,
how you have all these basically particles or cells kind of moving in a certain direction and you're tracing out that direction. So I mean, that's like I don't know how to describe it other than that, but it's interesting because that's not usually the way people think about it, and maybe because they don't have the data, they don't have the model. So this is real. I think this is really interesting. This component here. Um, yeah. So that's interesting. And then some of the things you found about post-synaptogenesis and some of these other stages in development are interesting. So you're able to kind of get this, um, kind of figure out lineage
yeah,
direction,
this
and then figure out some of the trends. Yeah, and some of the sublineages. I see that. Like, so in the ABA and ABP, it's decreasing, but in C, you have this fluctuating aspect to it. So that's good. I like that. All right, yeah, that's great. Did we have any other questions?
Um, yeah, one thing I'll just complete, I'll just begin the PPT, I'll just put all the 5 results and the conclusion and put the PPT in chat. And one more thing I wanted to confirm as to the way I'm thinking to build my proposal is in this way. I'll just share. I have some rough proposals here. So I was just, so I intend to produce in this way where I'll have an overall structure as this, like a paper where I'll have the contents and in this way. So basically, I'm trying to do in this way where I'll have the introduction first, then whatever datasets I'm using, the Bitfiller 2020 connectome dataset and the lineage tree, executive summary of what I'm doing. So if we see this, so it includes. So okay, this is a bit distorted, but yeah, this is the actual thing.
Oh.
So and this is the way in which I'm thinking to produce my proposal. So how would you recommend me to do it? So this includes all the work which I've done up until now. So and the justification as to why I did what I did and what are my trajectories, like what I aim to do in my future, like in actually the time of GSoC, what I aim to do. So this includes everything and everything mathematically grounded. So if we go to any section, we can see how exactly things were done and why they were done. So this is in a paper format, as if we can say it, like it's as if I'm writing a paper. So would you like it to be in that format, or would you like it to be a bit more informal or something like
Um,
that? Like, how would you like it to be?
I think this format is good. That it lays everything out clearly, and then I'd also like to see a paper on this, which would be great. And you already have it kind of set up, so
Yeah, yeah, sure.
it
I
can just be
also
modified
aim to
accordingly.
produce a paper on this.
Yeah, so that's good. Yeah, that's a good way to set it up. You just need to have at the end, well, at the beginning, you need to have like kind of an introduction to yourself and an introduction to the problem, and then the end you need to have the schedule, right? So that's what you need to add to it.
Yeah, yeah, right. That is missing. So this is up until now. This entirely is technical. Like all the sections are technical. But I'll just add timeline at the end. Is it fine if I add at the end? So here I'll add an introduction of myself first. then we'll go the whole of the technical part and then the timeline. Right.
Yeah.
Yeah. And one more thing. So currently it's like approximately 32 pages long. So is it a bit too long? Should I decrease it if this length is cool?
This is fine. Well, I mean, you can submit that as a proposal, I guess.
Cool then. Okay. So this basically includes everything, like whatever literature review I have done, what all like the graph attention techniques as well, and yeah, basically everything which I have done up until now.
Yeah, I think that would be good to see. Yeah.
Yeah, sure. I'll just send like it should be ready by tomorrow or after tomorrow. I'll send it to you for a review.
Okay.
That should be cool, right? Okay.
Yeah.
Okay, that's all from my side.
Okay.
Thank you.
You're welcome. Yeah, that's great. Thank you, Shreyas. That was wonderful work. If we had any other questions for Shreyas. All right. Well, there's Morgan. Okay.
No, really, really good. Yeah, looking forward to seeing more.
Okay.
Yeah, sure.
All right. Thank you. All right. So that's great. So it looks like Morgan and Jesse are here. Welcome. And speaking of Morgan, we had a presentation in our Saturday meeting, and I think he did a really good job of the project that he's been working on this past spring, and he was presenting on that in the meeting. And it was about taking all of the repos for the orthogonal lab, which are repos for some of the projects that we don't talk about in this meeting. And then we also talk about hypergraphs. So he took that repo as well. And he's working on building sort of an AI assisted tool to build models in JAX. So they were like simulations. And okay, hi Jesse. And so that was really interesting. So I'm going to go over his repo a little bit.
Okay, I just wanted to say I did find it, but it's 40 slides long. So
we can do that after this. Yeah. What I mean, you don't have to go through every slide, but
yeah, I'll just do it quickly through. Yeah.
Okay. So let's see. Okay, so this is Morgan's Organoid-hgx benchmark. This is he's doing this project where he's doing AI assisted building of simulations. So this is where this is his GitHub repo M9H, and it's Organoid-hgx benchmark. So Morgan is interested in organoids. And we talked about organoids in the meeting where you have these cultured systems of cells that differentiate and form different tissues. And then we can understand maybe things about the brain. So they form these kind of quasi organs or quasi tissues. And so he's doing this hypergraph for organoids, and he's got this code here where you have the benchmark data sets, you have data pre-processing. These are all steps in this pipeline. So he's building a pipeline. He's got the roadmap here. So the roadmap is, of course, phase one validation, where you use a standard benchmark suite. Then you have Pando reproduction, and that's just kind of some statistical evaluations. Then phase two, you have extensions, so you have higher order interactions. So you're looking at sort of the higher order interactions inherent in the hypergraph approach. And then dynamics, so you're looking at neural ODEs and SDEs. So these are ordinary differential equations and stochastic differential equations. And so this is the second step of setting up those algorithms and running them. And then, of course, we have Poincare embedding. So he's taking a cue from Shreyas, and he wanted to look and see if he could do this kind of in an automated fashion and build a model for the organoids. So that's where he's building and embedding, then looking at Poincare versus Euclidean on real lineage tree, and then validating the tree-like structure of self-fate decisions. Then he has the second data. So the first data set is just the standard benchmark suite. The second data set is the Pollen Lab CRISPR-I, which was published this year in Nature. This is Dissecting Gene Regulatory Networks Governing Human Cortical Cell Fate. So he's looking at gene expression data. So this is like different than what we've been talking about lineage trees and with anatomy. So we're looking at regulatory outputs of genes, regulatory networks of genes. And so this is where you have 44 transcription factors screened with CRISPR-I in primary human cortical cultures. This CRISPR is a technique used to do gene editing and things like that. Perturb-seq readout. So this is a next-gen sequencing technology, Perturb-seq. This is perfect for perturbation predictor validation. There's some data here on the bioarchive, and I don't know what the geo-accession is. I mean, we don't know what that is yet, apparently. And then, so this is just kind of getting this organized and in place. Then he has this cross dataset generalization, so train perturbation predictor on Fleck et al. Organoid data, test on Powen et al. Primary tissue data, and then evaluate transfer learning across culture systems. And then this phase 4 publication, so I guess replicating the publication figures, I guess is what he's doing here. And then being able to calculate the quantitative metrics, and then making this fully reproducible in a collab notebook. So this is like reproducibility now. So we're talking about replicating the figures in papers, making sure that they're replicable, that they make sense, and executing that on a collab notebook. So actually here he's using R and JAX. So R for the pairwise relationships, JAX for the higher order relationships. Then he has some scripts, Python scripts here, where he's doing these the benchmarks, the class balance ablation study, some of these other techniques here. And then he actually lists out the technical debt. So this is something that I like too, because I think technical debt is important to understand, because a lot of the even like a lot of the GSoC projects, we do them in like this 10-week period, and then they lie dormant for like maybe a year or maybe more. Like say you create a GitHub or you create a GSoC project, you work on it intensely for 10 weeks, and then you let it sit. And then maybe the next year, someone picks it up and works on the code. And we've been finding in our repos where we've been getting pull requests where things are broken, things need to be fixed, updated, etc. And so we know that like our GSoC projects suffer from technical debt. And that technical debt comes from just kind of working on it really quickly, making decisions that are like, okay, this works for what I wanted to do right now, and then letting it sit where it isn't, we're not maintaining it, we're not making it better, we're not going back and fixing things that maybe we took shortcuts on during the project. And so all those things kind of accumulate, and then the next time someone pulls out that repo and starts to work with it, you get a bunch of technical debt, and you have to fix that with a bunch of pull requests. And sometimes those pull requests overlap with one another, and they actually break more things than they fix. So we're actually having that problem now in one of our repos, where we're trying to figure out the best way forward, given that there is technical debt, given that there is things that people want to fix, actually testing it out on their machines, et cetera, et cetera. So I like this list of technical debt aspects, just kind of thinking about, okay, where am I in this project? What do we need to kind of create issues for so that people can kind of think longer term about these things? Let's target things that can be repaired. Let's target things that can be thought out in more depth. And let's target things that we can maintain, like maybe come back to every 4 or 5 months and revisit. So this is the list just for this repo of these things. And of course, when you're working with data sets, you get a data set and you process the data set. And sometimes the data set isn't perfect. In other words, it doesn't do exactly what you want it to do. So sometimes you have to make decisions about how to normalize the data, how to use it in conjunction with other data sets. And people, sometimes you have to like write that up very explicitly. And if it's not written up very explicitly, people have problems replicating it or using the project. So I just like the way he's approached this, both from the technical debt standpoint and from kind of thinking through these data sets and how they're used. And you can't be too descriptive here because and this happens with documentation as well for projects. A lot of times the documentation is vague or non-existent, but also with data sets, it's a tradition in science to be very precise about your methods so that people can replicate those things later. And with like if we're using AI or even if we're just using interactive notebooks, we can just build that right into the notebook or to the GitHub repo, and it's automated in terms of replicating things. So that's a great advance, but still we have to keep them in mind that sometimes if they break and we just kind of rely on the automation, that can be a problem. So that's another thing to keep in mind. So it looks like Morgan has configured Claude for this, and this is Claude just kind of as a contributor to the repo. Yeah, I like that, Morgan. Thank you.
Oh, thank you. Yeah, it does have a focus. It does have a focus on the gene regulatory networks in control of cell fate. Like it's not exactly matching. But as you know, I am trying to be doing some organoid work, even though we're still more at the neuronal culture stage here. But yeah, this is just an exercise in agentic coding, with some thought to how you can do this in a kind of practical and what's the right term? Well, just trying to learn best practices in terms of how to be generating code that you can still test and maintain.
Yeah, yeah.
And I had the my first real job out of college was working on software engineering for a medical device company where there's a certain amount of regulation in terms of how that code is made and documented. And I've as well as the fact that like a lot of the code that we were using was originally lab code.
Yeah.
Right? So it was taking over code that had not been developed in a regulatory environment, and yeah, so so definitely trying to think of some of those things. Think about some of those things. What I learned is that if you want to have sustainable code, if you're like code that lasts beyond the original author,
Yeah.
that these kinds of practices are just absolutely essential. And you never want what Microsoft called to have software an understanding of your code base transmitted by the oral tradition.
Yeah,
I think
right.
is how they put it. Yeah. And so right. So just trying to think yesterday, going over that because again, like for that presentation, Claude also wrote the presentation.
Yeah.
But I was trying to think about how to track the software product itself in terms of like lines of code but also other measures that get at the the amount of code coverage, the amount of documentation that covers that code, how to and kind of like how to capture all of that and monitor that as an output, because again, I feel like this is something that we're gonna have to learn how to how to produce code differently,
Right.
but it will still have a core set of kind of principles that we have learned from the software engineering practices of that have grown up since the 60s.
Yeah.
And I thought it was also really interesting going through this regulatory software, the practices, that how much of that was actually based on, building big systems like airplanes,
Oh yeah.
where there's a lot of what they had was a lot of systems integration issues, and so for some of these like more complex products where you've definitely got multiple authors, so to speak, um, holding them to a kind of set of interfaces, as well as like when something goes wrong, are you able to trace where the problem arose, you know and that means both in terms of the software deficit, as well as like when was it introduced, and when and where was it introduced, so it's yeah. So that is kind of new, though, in terms of what kind of metrics should we be producing to capture this auto-generated code, and potentially auto-generated unit tests, auto-generated code, auto-generated documentation. How can you produce 10,000 lines of code and still have it be a kind of digestible, an understandable construct, or an understandable artifact by a human, right? And yeah, so it's trying to think about these things. And I thought I might have pushed this yesterday, but the HGX, the hypergraph library that was actually like the real product of the organoid work.
Yeah.
I had some great benchmarks. Basically, I was benchmarking it yesterday against kind of like the leading PyTorch libraries, and that was cool. I wonder if everybody's just a little off of the time zone.
Oh yeah.
And we're getting more people because of it's 8 o'clock. Okay. Yeah. It's so the cool thing being that this hypergraph library, again, I want the hypergraph library to be actually a contribution to JAX, right? And the question I'm trying to have is like how to make it, how do you do that and make it something that's that somebody else, make it worthwhile, so that somebody else doesn't just spend the day having an agent generate a new library.
Yeah.
Right. So kind of like beyond just so the performance is the nice thing being that against these PyTorch libraries, I'm seeing quite comparable accuracy, and I'm seeing in some cases almost 100x speed up. And this is, I'm running on a Blackwell. I'm running on a DGX Spark, which is an example of a very new chip from NVIDIA. But I mean, I'm running these PyTorch in a NVIDIA GPU container. I'm doing it the NVIDIA way. I'm doing it the way NVIDIA tells me to run PyTorch on this. So I don't feel like I'm trying my best to give PyTorch every advantage, but the performance looks great, but that's not the value add, right? So or at least I think it's going to be more than that, right? So yeah, we're still learning about this, and really interested if people have thoughts about how to approach this. So.
Oh yeah. Well, let's talk about that in the Slack. It's really interesting. Great, thank you. I wanted to highlight one more thing, and then we'll come back to Susan. This is Hardik, who has this neural connectome growth simulation. He actually put it in the Slack. I just wanted to highlight this. So this is where we have this neural connectome growth simulation, simulating how neurons grow, connect, and form complex networks using biologically inspired rules and graph convolutional network embeddings. So this is an overview here where he's running this simulation of developmental growth of the neural connectome using a 3-layer graph convolutional network and a self-supervised approach to compute a 16-dimensional embedding vectors for each neuron, capturing both structural topology and spatial identity. So we're getting a lot of that kind of work this year. So we have growth rules here. So we have neuron birth, preferential attachment, spatial proximity, connectome formation. So neuron birth is the birth of a node. Preferential attachment is how it attaches itself into the network. Spatial proximity is the sort of how likely is something how likely are two nodes or two cells to interact or connect. And then connectome formation is where you get synaptogenesis, or those connections kind of refined. And then that's kind of how this works. And so then he has a project pipeline here, node features, the GCN input, the GCN architecture, and then the key results. So this is a nice repo as well. This is Hardik Singh 8, and it's neural connectome growth simulation. And the interactive notebook is here if you're interested in running this. So it's like, you have to open it in Colab. You have PyTorch written, so you can, kind of run that on your own. All right. So Susan, are you there? Oh, you're muted.
Yes, I'm muted. Everybody probably wants to present their projects, but there's a time zone difference.
Oh, okay.
That's occurred. Anyway, if you'd like me to try to share my screen, I could do this, and I'll run through it fast.
Okay. Yeah, that's fine.
How about that?
Yeah.
All right. This is my thesis presentation. So I modeled simple epithelial tissue using tensegrity structures with difficulty. And so this is the outline, this is the introduction, and I showed some of this last week. These are epithelial tissues, the different kinds of epithelial tissues, and I used the three simplest, squamish, cuboidal, and columnar, to model. And this is somewhat what I was looking for. This is a stress strain curve for skin. And this is stress here and strain. And stress strain curves are sort of an engineering standard. So I showed this. This is the setup for a cell, an epithelial cell. It has a belt on the top, usually on the apical side here. That's a tight junction and an adherence junction. And they hold the cells together along the apical surface. And to go with that, there's actin and myosin stress fibers in the belt that goes around the top of the cells. And it is this non-muscle myosin, but it still acts like a muscle, and you get ratcheting behavior where you can constrict the cell's circumference. And then the cells are held together with trijunctions. This is an actual image of the trijunctions, and this is the model. And this is a close-up look at the molecules involved. And I modeled my cells just exactly like this. Hexagons held together at trijunctions. So this is my model. So I had a top ring, and then I had the trijunctions, and then I had the basement membrane attachments. So model build, I started off with these. These are T3 and Tensegrities. And I use these for the trijunctions. And I also use these for the trijunctions. These are the trijunction in the middle of the cell. And these are the trijunction on the edges. And if I modeled it any different way than that, it didn't work. Like the program said, the building fell down. It was that type of program. It was, I don't know, struts and strings. So this is a bottom layout, just a hexagon. And then these are the sides. I used these crossways links to build the Tensegrity. And then the top layer, there was a top layer, and I use it going the opposite direction, the cross link going the opposite direction to the bottom. And if I did it any other way, it didn't work. So here's the single cell hexagon structure. And you can see that there's a strut or a bar going this way and another one going the other way for the top ring. And then these are the junctions. So I have the T4 complicated junction in the middle and the T3 on the outside connected to these just scaffolds to hold the whole structure up. I wanted to put more cells in here, but and try it out, but I ran out of time. Yeah, so there's the structural detail. There, the trijunctions are quite a bit smaller than the cells. And here are the three types of cell models I used: the cuboidal, the Squamish, and the columnar. And boundary conditions, I have put spring foundations on the bottom edges of most of the cells, except where I had to attach it firmly to the substrate. And these are the pinned positions. So I only had 3 pinned positions, and the model did work with that, which is good because the substrate's an important part of the cell's environment. The ECM, or extracellular matrix, is an important part of any cell's physical environment. And these are the boundary conditions I use. These are in newtons per meter, and the forces are in nanometers. So I got 200 nanometers, and I was getting that data from atomic force microscopy papers. And then I applied the force just from 0.1 nanonewtons, pardon me, to 200 nanonewtons, and it's in the middle of the structure. And this is how I applied the force, and it's to this T4 trijunction. There's the three arrows showing where I applied the force. And then it was applied over this area, because this forms a triangle up here. So this is the area that I applied these three forces to equally. And then I measured the engineering strain. And more or less, this is, I had the hexagon 3-cell model, and I modified the top and bottom to try it out. And I changed the forces, I changed the pre-strain, and I changed the spring foundation. Results. So overall observations, cytoskeleton values are important to this cell's mechanical environment, and the cell-to-cell connections are very important, as well as the cell-to-basement membrane attachments. And that's what other people found. And this is just some graphs. So these are graphs in tension and graphs in compression. And it's opposite to what a normal person would do, because they would put tension here and compression over here going down. This is a graph that goes down. But anyways, this is a standard engineering stress strain curve. Oops. And I'll go back. What I was looking for was this separation of curves. See there, you can see that they are separated, and this gives a cell a control over its environment, basically, mechanically speaking. And I've discovered that there's a value of K that's important. So this is the elasticity of the string times its area over the elasticity of the rod times its area, gives you the K value. And this changes the slope of the curve. So I was able to match my model to a variety of tissues. Anyways, and this is a cell connection. So if you've got the upper ring, if it's at 0.12 micrometers or even 0.1 micrometers, you still get a separation of curves, a decent separation of curves. But if you make it too small, if you make the distance between the cells, or the height of if you take the distance between the cells and make it too small, at 0.01 micrometers, then the curves collapse. So the distance between the cells is important as well. That was a discovery. And then this is cell to basement attachments, and this is just a sample of a very nicely separated curve set. I'll just carry on here, and this one shows it's basically collapsed. Like this is not a good situation for a cell to be in. It can't manipulate its environment at all. Anyways, I then matched my curves to live cell sheet curves, and I have, this is my model in the dotted lines here. And this is what they got for this cell sheet. And the cell sheet was laid out in a random fashion and then a more organized one by putting grooves in the substrate. So this is the most organized one, the OCS-5. And if you look at this, the OCS-5 matches my curves when you're measuring the strain on the cell sheet perpendicular to its grooves. So I did get a matching graph, which was nice. And this is all in tension. And then I tried compression, and I did sort of get a matching curve. This is for lung. Lung's complicated. I don't know if you could exactly call it an epithelial tissue, although they do, but it's complicated. Anyways, I did get kind of a matching graph for this. And this is more complicated than that, but I'm just going to carry on. I managed to kind of match where skin was at. So I have again the dotted line here and the dotted line there from my model, and they kind of match what was going on with skin. And this is simply by changing that K factor. This is K equals 4 times 10 to the minus 4. And conclusions, the cell model worked, and I was able to match tissues to it. And I was able to use my model past 20% strain, which other models are not able to do. So this is a good model for if you wanted to cover the full range of tissue, because it tends to stretch over 70% sometimes, not up to 10%. Conclusions, yeah. Anyway, questions?
Well, that was good. Thank you. Yeah, so that last slide, the hexagon 3 model, you got good performance relative to what? Like just different models in the literature?
Yeah, different models in the literature. The Ogden model, for instance.
Okay, Ogden model, like
Ogden, yeah.
Okay.
It's often used for this. The hyperelastic models, I got agreement.
Okay.
Well, I went beyond what a normal hyperelastic model does, and that's modeling the stress and strain in a tissue using springs and dashpots.
Oh, okay, yeah, springs and
Continuum
dashboards.
model.
Yeah.
So my model was better than a continuum model in that it covered the full range of tissue.
Okay.
All right. I'll shut it down then? Good?
Yeah.
Okay.
All right. Yeah, that was good. Thank you, Susan. So I'm to understand the Tensegrity is outperforming continuum models and like spring and dashpot. Yeah.
Yeah, even though it was difficult to model with the finite element analysis because tensegrities tend to move more. They're otic and they tend to move more than say a building does.
Right.
When it's yeah. So it continuously, I believe it continuously told me that the building was falling down.
Oh,
so
yeah.
actually,
Yeah.
I had to go with a very stable structure. It's a very stable tensegrity structure.
Right.
Yeah. I like to investigate it more, but anyway.
Yeah.
I graduated last year and just done with trying to pound a computer over with my forehead.
Yeah. Okay. Well, thank you. Yeah.
Okay.
So, yeah. So it looks like we have a bunch of people here joined at around top of the hour. So I don't know. Looks like Varshith is here, Mehul, Utkarsh, Janaksinh, Barshan, Ashu. So, Mehul, I haven't seen you in a while.
Yeah, hi Bradley, hi
Hi.
everyone. So I joined last week and we were listening to the presentation, today's presentation only.
Yeah.
And there were a bunch of folks like who reached out to me, and I just like asked them to join today's meet so they can present and take feedback like from the community as well. So a bunch of folks I was talking to, Shreyas is there, Ashu, Barshan, Janak, and I think there were more I was talking to. So yeah, I just wanted to like hear everyone like for the introductions and the questions they have.
Okay. Yeah, thanks. Varshith, are you there?
Yeah, hi Bradley, how are you?
Hi, how are you? Did you have anything? Yeah, report?
No, nothing right now, but I have done the changes which I have said for our paper. I've done the changes for the reference. I'll send it to you in 1 hour, I guess.
Yeah, send the email. Yeah.
Okay.
Thank you. All right, so we have a lot of people here on the who want to present, but we don't have a lot of time. I just wanted to kind of maybe hit a 5 minute at most, a 5 minute report from each person, maybe less than that.
Can I present, right?
Yeah, go ahead.
I just had some like really good results which I'd like to share today. Is the screen visible right now?
Yes.
Yeah, so you might remember I was proposing agent-based models where a cell can be considered as an agent which has an internal state that responds to its environment, its neighboring agents through physical and chemical interactions, right? I actually went ahead and built one. Let me just quickly go ahead to. This is the like simulation that ran.
Oh wow. Yeah.
Yeah, and it looks amazing because let me just tell you what happened here. So first of all, these agents had their internal state as position, volume, meanest identity, their physical contacts, addition, cortical tension, and like the ellipsoidal eggshell. Out of these three terms, like the cortical tension, addition, and volume elasticity. Volume elasticity is determined by the gene. Apart from that, the cortical tension and addition, I'm trying to discover them by generating the graph itself through the like base principle. So like take this for example. Like I have 5 different energy terms, and like I wrote them myself. For one represents the force that the actual like forces on different cells to keep them inside the boundary. Another one would be like the incompressibility of individual cells that prevent them from shrinking, and one that prevents 2 different cells from overlapping with each other. The JKR law that like finds the area of contact, and I can calculate the addition energy based off the individual cell, right? Then I have the like PR protein, which dictates the cortical flow, and like which is like basically responsible on how the cell would divide into different axes, right? So what I'm doing is I'm trying to find the values for these like the cortical tension, addition, and KV such that these energy terms are minimized. So I'm basically trying to find the biological like equilibrium. I am actually running 2 different optimizer loop. The first one is basically an STD, but with the momentum 0, since inside cell the Reynolds number is like really small, so there's no inertia there, right? Then like let me just talk down the technical terms and like explain in simple terms, right? So the whole problem is we have a real embryo contact areas, and we want to find the cortical tension, addition, and alpha here representing the like the difference between the recorded volume. It was in voxel units, and in my simulation it was millimeters. So the alpha term is here for that. So the inner loop which is the physics simulation. I am trying to, like, given fixed set of parameters, I'm trying to run the simulation and letting the cells find out where they should naturally settle, like dropping marbles into a bowl and they roll to the bottom. So we can say we have 820 steps of rolling downhill to the like minimum energy. Like PyTorch calculates that through the gradients. Then there is another rule for that, which basically calculates the other parameters, the physics parameters. So after the inner loop finishes, I compare the predicted contact areas, pairwise contact areas for different cells with the real C-shaper areas, and then I compute the loss. I match the different parameters slightly, and then rerun. This is slightly inefficient because right now I'm just trying to figure out the values by random guessing, I would say. Like I just increase them by 15% at a time and see whether the loss decreased or increased, and I change it according to that. But like these are the results that I got. So here are the values for like the cortical tension of AP lineage, EMS, and the P lineage. These are pretty close. And here you can see the predicted contact areas versus the actual ones. The R squared was 0.38. So why I was modeling it like this way, there was a very specific reason. Like I was pushing forward towards the white box model instead of the neural networks, right? I was trying to answer the question, like if a cell moves to this position, why is it moving that way? So I figured out some problems like 0.38 was the hard ceiling and not a tuning problem. Like no amount of training would fix that. The reason was that right now my cells are like spherical units. These are not the actual vortex models. That is the problem right now. And I even got greedy and tried to push forward like the 8 cell, like extend it to 8 cells, and it failed. I had to like recalibrate all the values, and the only successful run that I got had the addition to 10 and alpha to 10, which are both upper limits, which is not like really helpful. These are like the absurd calculations. There are also some problems like I had the K shell and K repulsion, which were like hard set by me as regularizers and not something that were derived like biologically. My inner loop, which I was talking about here, like this point, which calculates the gradient to minimize the to find the optimal point for the position like the XYZ. It is purely deterministic, unlike the actual biology where cells like vibrate around to find the best best set of position, right? So I actually read some more things and I found out there's like Cindy. Cindy is basically a machine learning algorithm that finds equations through these set of trajectories like given points. So basically, if I take some set of data, so I have some snapshots at different time and I provide them to Cindy, then it will map out some equations from that will give me exact like reasons for movement that I was talking about, which was my like whole goal. But since the C-shaper dataset doesn't have that many observations, like it only have two timestamps for 4 cells, like the early embryogenesis. So I ran Cindy on my own data, but like I got the results. These equations you can see like minus 0.0321 the cortical tension plus 0.0102 NN. NN is like neighbors, N neighbors. And like these represents the velocity terms. These are telling like which direction the cell would move to based on the cortical tension and the like neighbors. This is like really helpful if you are doing a research in this specific area. Like you can understand that these two are the only terms that are playing role in cell movements. At least like I can say that based on the 25% like the 0.25 R squared value. But I also previously explained like there was this problem in this entire architecture. What if there are some parameters that we are unaware of, like what if there are some other units that we haven't figured out or discovered yet as humans ourselves? Then we have to shift to GNNs, because like GNNs figure out parameters based on the data. Like they build the weights around around the parameters. They figured them out themselves. So I had this amazing idea. Like what I'm proposing is that we have another layer of GNN and we calculate the difference between what GNN produces. Like we consider that as the maximum output, like the maximum data ceiling, the maximum information we can get from a data, and then we compare it with my model of ABM, and that would basically give us like if there is something missing. For example, in my case, I know that vortex model is missing from my exact like the data set right now. This is it that I was doing.
That looks great. Thank you. Yeah, I like some of the things you're doing with agent-based models and with discovering equations and then figuring out where that fits into biophysics and then finding the boundary of GNNs. Because the GNNs, we're working with data from, limited data. And so we'd like to find out like maybe a larger universe of possibilities. Yeah. Thank you.
Yeah.
That's great.
Actually, like I was facing a problem with GNN. So like the problem was that there isn't enough data to train the GNN model on.
Yeah.
It was previously available, but I don't know. I checked it last week, and I guess they removed the access from the SharePoint for C Shaper.
Oh.
Can you please check once and maybe inform? I'll ask you on Slack.
Oh. What is that C shaper for? What is the access problem?
I was like I previously checked that on SharePoint, like the C Shaper dataset for contact areas, their XYZ points and their volumes. I was using that previously, but like I checked those last week and I couldn't access them anymore. I thought maybe like somebody removed them.
Is
I'm not
this
sure.
who's managing that SharePoint?
I'm not sure. Let me just check it.
Oh, you just found it online and then you downloaded it, or?
Right, yeah.
Okay. Yeah, I don't know. I'm not familiar with that data set.
I'll share that on Slack.
Yeah, that'd be good. Thank you.
Thank you.
All right, thank you, Utkarsh. Barshan, do you want to talk for about 5 minutes?
So, am I audible?
Yeah.
Okay, so I'll just give an overview. I didn't prepare a presentation because I was most going through the documentations of the various tools I'll be using.
Yeah.
So I prepared documentation, so how I can incorporate that into understanding the attention scores. So yeah, there is a like tool for it. It's API, so that is doable.
Okay.
And other than that, I was looking through my proposal and stuff, and also I created a repo and created a initial code base with the datasets. I downloaded the dataset from the Epic, so there are like a lot of data over there, like of different employers. So that should be enough to train a deep learning model.
Okay, that's good.
So
thank you.
Get the initial setting up the base basically before I start coding.
Oh yeah, definitely.
Yeah, that was all. Thank you.
Okay, thank you. All right, Ashu, would you like to go?
Yes, sir. Am I audible?
Yes.
Okay. So, firstly, I would like to start by introducing myself. My name is Ashu Rajput. I'm from Lucknow, India, and this is my first meet that I'm attending here. So, I will start by telling my planned approach. Like, this is the series that I have made. First, the development data set. Here, the raw biological data will be formatted and scaled like only the good features will be selected in this. And then next, the temporal graph construction. Here the biological data will be converted to network X graph. Why we will convert it to network X graph? Because it is ideal for constructing graphs. Like it will add nodes, graph, compute graphs, properties, and visualize. Then after this, we will convert it to PyTorch graph, PyTorch geometry graph, because graph neural network requires tensor format. For this, it will add edge index, node features, and labels. And then the graph neural network will be shown. So why it help us? Because the edge construction, it divide it into phases like linear edge, spatial edge, and temporal edge. In linear edge, the mother-daughter cell will be separated. In spatial edges, spatial interaction will be constructed using nearest neighbor algorithm, and which include the k nearest neighbor and radial graphs. And for temporal edge, it will see that temporal continuity will be modeled by connecting the same cell across time. Like the cell, suppose cell A, and the next for time lapse, it will be added. And for node representation, it will take the node ID and time. This I have shared you the visual representation of this that I have tried using a data set. I was just testing it. I think I should show you my screen. Let me just share this. Actually, I was trying to test some random data set, and this is what I have constructed. This is for embryosis, like cell division.
Right. Okay, that looks good.
I was just searching for some data set and I found one, so I tried to implement this.
Yeah. Oh, that looks good. Yeah.
And so for development graph neural network, like I have planned to use this because the development graph neural network is a model that learns how neural connections form during development. Like from node features, it will make node encoder MLP, multilinear perceptrons, and it will add a feature like features have different scales and formats. The node encoder converts them into dense vector embeddings. Like suppose if a node is having type this x, y, z, and h. So after this, all the nodes will be converted into a similar dimension, 64-dimensional embeddings. It will help because the data features are sparse, and it will create some type of noise while creating a graph. So it is better to convert it into node encoder. And then comes the graph attention layer. So what it does in the neural network graph, not all neighbors are equally important. Like some carry more weightage than the others. So this layer is important to just know which neighbor matters the most. And it has weight matrix like which will tell the importance of a node. And then it will be then the temporal memory development happens over time. GRU, the graded recurrent unit, learn time dependencies. This lets model remember previous development stages. Like it is that it will help remember the past stages that a node and edge has passed, so that it creates more realistic models. And then the edge prediction comes after all this, after which, and next part is the emergent connectome generation. So it like after this, the weightage of a node and edge is find out, and the previous development stage is known. Then it is better to create a connectome between nodes. So I have also made a simulation of this. I'll show you. This is also I have tried on some random data. It is not a specific data type.
Oh, that's good. Yeah. Great. Yeah.
So I have tried it on some random data.
Okay.
I will surely be using a good data. And for hypergraph modeling of neural interaction, there are two main libraries, HyperNetX and Deep Hypergraph Library. So these are the libraries which are good for hypergraph modeling because it building hypergraphs and analysis, hypergraph neural network and learning patterns. So here is a code that demonstrate how it is done. So because in hypergraph, the edges connect to more than two nodes. So the hypernetics, there is an import hypernetics as HNX, and these are the edges and the nodes that it is going to connect. And this, you have suggested me to see Krill's presentation, and I approached him and see his repository and presentation. In that, he majorly focused on the first cell division and by protein formation and centrosomes. He has shown this presentation where two cells were merging, centrosomes were emerging, the protein formation and all. So what I have taken reference is we can see neural birth, neuron migration, axiom growth, synapse formation, and microcircuit emergence. This we can implement by that reference, what Krills has presented in his presentation. It was majorly embryosis growth. That too, it was for one cell division, and it took 18 minutes for that cell division. And in C. elegans, it usually takes 13 hours to fully divide itself. So what I have thought that it would be better if we use that simulation for this pipeline, because from 0 to 100 simulation, it will be very computationally expensive. So the simulation components for this include realistic and 3D neuron objects. It will use position of neuron, velocity, axiom direction, axiom length, neuron type, age, and connections. And for axiom object, represent the growing neurite, start neuron, growth direction, growth speed, branching probabilities. And for synapses object, representation connects presynaptic neuron, postsynaptic neuron, synapse, and strand. It would be better, it was computationally very expensive, so it is something that can be made, but I am still doing my research on it. And the physics of this simulation includes neuron migration, axiom growth, synapse formation. This was about my presentation, sir. And sir, I want to ask one thing. I have a question.
Okay.
While my research, I come around this research paper that I think will be can be taken reference for the last simulation part that I have shown you for neural birth, this thing.
All right. Yeah. Yeah, that's from the OpenWorm group. We're affiliated with the OpenWorm group. They use C302 for modeling their connectomes, and that's something you could. I don't know if you'd be able to fit it in easily with graph neural networks and that, but it's a method for doing that sort of thing. It's basically handling the data in neural ML and then plotting out the graphs and that sort of thing. So you could pursue that. I mean, you could look into that a little bit. It would fit in, I think, with a lot of the things going on in OpenWorm, so it'd be useful.
Okay, sir, I'll take my research and I'll let you know. And sir, I have shared my draft proposal to you.
All right.
If you could just review it and give suggestion, that would be very helpful.
Okay. Sounds good. Thank you.
Any feedback, sir?
Yeah, I don't know yet. We'll see. All right.
Okay, thank you, sir.
All right. Thank you. Why don't we call it a day? Oh yeah. Well, yeah, we had a time zone change like 2 weeks ago here, so yeah, I have to go, so we'll continue the conversations in the Slack, and we'll be here next week, of course. People can present then if they want. Okay, thank you. Have a good week, and good luck with your projects.
Thank you.
Thank you. All right. Talk to you later.
Bye-bye.
Bye.
Thank you, sir.
Thank you. Bye.